Hello, everybody. The topic of my talk is about a new approach to study communication under realistic conditions by using identified nerve cells of insects and their activity to explore hearing in nature. Let me introduce the problem by using my favorite insect group, which are grasshoppers, crickets, or katydids as an example. Acoustic communication between a male and female katydid appears to be a quite straightforward behavior. Usually, the male produces an acoustic signal, and the female, once perceiving and recognizing the signal as species-specific, shows a response, often a movement towards the male. But the system is not as simple as it looks like. Communication does not happen, and of course did not evolve in a soundproof room, but in a physically complex environment where the transmission channel for the sound may degrade and attenuate the signal, and the channel may be noisy. In my talk, I will focus on this aspect of the transmission channel. To study sound communication under these conditions, we developed a new method to record the activity of auditory nerve cells of an insect directly where insects communicate, an approach we called biological microphone. We gave this name to a portable preparation. You see the insect is mounted on a holder and one or two electrodes are attached to identified nerve cells, which we know in great detail from lab experiments. All the electronics necessary to record action potential activity are in this socket here. And it is so stable that we can place a preparation at any position of receivers in any habitat, for example, in the tropical rainforest or in a simple meadow. And we can record and listen to what the animal is hearing through the ears, which are located in the foreleg here. So this is a realistic way to study perception for the nervous system. The result is, for example, an action potential activity like this one, consisting of spontaneous spikes, bursts of activity, repetitive activity, and single bursts. And based on this activity, the insect brain must make important decisions, for example, to approach or to avoid a sound source. And as you see, this is not an easy task, because only we as performing the experiment, we knew that this and this was the result of bad echolocation signals. And only we knew that this small burst of activity was a response to playback of conspecific song. So with this approach, we could study, for example, the important question, how well a signal is being represented in the nervous system and over which distance. And as you have seen, because a signal can be distorted and attenuated on the way to the receiver, this is by far not trivial. So here we did broadcast a repetitive sound signal with a speaker, and at the source, the signal looks like we designed it. But at a distance of 10 meters, you see that these sound pulses are heavily distorted, and no single sound pulse looks the same at the receiver. So using our biological microphone approach, with a nerve cell responding to the sound, we could get direct information about the reliability of sound perception under these conditions. Here we broadcast 10 repetitive sound pulses. Again, with the microphone, you see strong and different attenuation, and we see that the bursting nerve cells shows high variability to each of these sound pulses. And more importantly, you see here that the microphone signal is almost lost, but the nerve cell gives a strong response. So in this case, the preparation and the microphone gave us different results. When we switch the signal from a pure tone at 20 kilohertz to a broadband signal with a mid-frequency again of 20 kilohertz, we see that a lot of the variability is gone, and the nerve cell shows high reliable response to each of these sound pulses. So we can conclude if an insect signal uses instead of a pure tone, a broadband signal, this gives a better reliable perception over distance. And we could indeed show that for this katydid, reliable signal perception is guaranteed up to a distance of 40 meters. However, various signals in a broadband signal do not attenuate the same from sender to receiver, but higher or ultrasonic frequencies in a complex signal suffer from stronger attenuation than lower frequencies. Let me illustrate this point by using field data of another katydid, which we recorded far north in Canada. This insect uses a strange signal, which is a repetition of an audio mode with frequencies below 20 kilohertz, and this is switched predominantly to the ultrasonics at 34 kilohertz. And this repetition goes on sometimes for hours. So we wanted to know how these different modes in the signal are being represented at various distances. Again, we used our portable preparation, and I show you just one result at a distance of 10 meters from a singing male compared to 2 meters. For your convenience, I marked the audio mode here in red, and you can clearly see that at a larger distance, the neuron does only respond to the audio mode, although the neuron is equally sensitive to audio and ultrasonic frequencies. However, at a shorter distance of 2 meters, the neuron now does respond even better to the ultrasonic mode, as you can see in various instances here. So this frequency-dependent attenuation can be used by animals to judge the distance from a signaler. And in a follow-up study with this insect, we could indeed show that they use this excess attenuation for ultrasonic frequencies to maintain a distance to other signalers. Another problem for hearing outdoors is directional hearing, because in almost all cases, a receiver must know not only that there is a sound, but where the sound is coming from. This can be a problem in scattering vegetation, where sound waves arrive with almost equal intensity from all directions. In other words, directional information can be, in the extreme case, completely lost. The approach we use to study directional hearing under such conditions was to record simultaneously, as you see here, the responses of two interneurons, one on the right side and one on the left side. We knew from lab experiments that these interneurons were directional sensitive. And in this case, we started with a receiver on top of the vegetation and presented sound coming from left, from frontal, and from right. And you can see under these conditions, sound from left results in the left interneuron responding stronger and sound from right in the right interneuron. However, when we place the same preparation at the same distance on the ground, such directional information is lost, although both neurons do respond to the sound. So we have the clear case that sound perception is still working, but directional hearing is not. So we can conclude that these directional differences appear within scattering vegetation. So directionality of the auditory system depends not only on the physical properties of the ears, but very strongly on the spatial configuration between sender and receiver. A final problem is noise on the transmission channel, which means that not only the signal needs to be detected and discriminated and localized, but it must be detected in the noise of other sound sources. And these are often other communicating animals. This is most dramatic in tropical rainforests, where after sunset, many different species of insects start to communicate with their calling songs, which gives a cacophony of sound with a mean background noise level of more than 65 decibels. So how can these different species and individuals communicate and hear in this complex situation? So here you see the noisy situation in the upper panel. This is a sonogram in the frequency range from 1 to 9 kilohertz. In the tropical noisy rainforest of Panama, we counted up to 53 species of cricket, and all calling within this relatively narrow frequency range. The cricket we studied was this cricket with a calling song in the lower frequency range here. If we present this noisy situation as an oscillogram, as shown under B here, you see that the overlay of all these sounds produced an undiscriminated background noise where no amplitude modulation is visible, at least not for the par eucanthus calling songs. Again, we used our biological microphone and placed next to the microphone which recorded this song a preparation of an interneuron which is called AN1, and we know from many lab experiments that this interneuron AN1 is responsible for presenting the conspecific calling song with its spike activity. You see all this bursting spike activity. The assumption was that under this noisy condition, the tuning of this neuron must be very high in order to represent only the conspecific song. And indeed, if we filter this unspecific noisy situation with the tuning of this AN1 neuron, we suddenly get a clear amplitude modulation. And as you see, this amplitude modulation corresponds nicely with the bursting activity of the interneuron. In other words, despite the noisy situation, we have a solution by selective filtering which guarantees that sound perception of conspecific song is possible. So I hope I could convince you that the study of sensory coding directly under conditions where communication takes place can give us new insights into the way communication happens. Thank you for your attention.